would like to introduce everyone to Dr. Anthony Bogdati, who is our resident Western blot and protein detection product manager and our Western blot guru. And he will be discussing the new Odyssey M imager from Lycor. So enjoy.
Hi, everyone. Can you guys see my screen?
Yep, we can see it.
Okay, perfect. The right screen. All right, so yeah, thanks, Fraser. So as Fraser said, my name's Anthony. I'm the product manager for Lycor at Millennium Science. And so today, what I'm going to do is talk a little bit about Lycor's newest addition to the Odyssey family, and that's called the Odyssey M. So the Odyssey M is the most powerful Odyssey family imager to date. And it's really well equipped to handle even the most diverse protein expression studies. And this is predominantly due to its versatility as a system. So just now, stop on the next slide. All right, cool. So just a brief overview of what the Odyssey M can actually do, or what it actually has. It has over 6 logs of dynamic range. And what this means is that you can detect both low and high abundance proteins in a single image, single acquisition, without any saturation. And it has a lot more channels compared to its predecessors, the Odyssey XF and DLX. It has actually 18 channels compared to 2 or 3 channels for those aforementioned systems. It actually has 9 fluorescent channels controlled by 4 lasers, 4 epifluorescent channels, 4 transfluorescent channels, and 1 chemiluminescent channel. It has the ability to do much faster scans than its predecessors, and this is primarily due to the fact that it uses a line scanner compared to the point-to-point scanning system of its predecessors. And this really allows for a much faster scan time of around 40% faster scans. And another great thing about it is it has a much higher resolution for imaging tissue sections, for example, up to 5 micron resolution compared to the 21 micron resolution from its predecessors. So really, this all culminates in the ability for the system to do many more assays. I like to say infinite applications. And you really do have a wide array of imaging options with the system, where you can do anything from infrared fluorescence, visible fluorescence, fluorescent proteins, bioluminescence, chemiluminescence, colorimetry, H&E staining, and cell health assays, and so on. And this is really just the tip of the iceberg. There's a lot of things you can actually do, any imaging, essentially. Now, today I'm just going to take you briefly through some of the possible applications that we can run on the Odyssey M, with a bit more of a focus on fluorescent Western blots, because these are the most popular type of blots that you can do. So in terms of the supported applications, this is just a screenshot from the actual software that runs the system. So it's sort of broken up into membrane applications, gel applications, multi-well plate applications, and slide applications. So starting with membrane applications and everybody's favorite fluorescent Western blots, you can essentially do, so we have your 800, 700, 520, and 488 channels. You can do your typical 2-color Western blot in the 800 and 700 channel. And additionally, you can do your total protein stain in the 700 and the newly added 520 channel using the newly added revert 520 total protein stain. And of course, now the system has the ability to do, excuse me, 3-color Westerns with the addition of the 520 and 488 channels, and the addition of the VR dye 520 and VR dye 490 antibodies. So before I talk about 3-color Western blots and whether you can actually do them, I first want to talk a little bit about fluorescent sensitivity. So if we look across all the channels, you actually can see that the 488 and 520 channel does have a lot more background than the 700 and 800 NIR channels. So the important thing to note here is that you can kind of think of the near-infrared channels as those premium wavelengths. And you're going to preferably use these for your low abundance proteins, the ones where you need the best detection, and sort of keep the 488 and 520 channels for your higher abundance protein. And the important thing to note here is that although we have a higher background in 488 and 520 channels, the important thing is that linearity is not actually compromised at any of these wavelengths. And you can actually find a combined linear range across any of these wavelengths. And that is super important when you want to multiplex for 3 targets with 3 antibodies. Again, as long as you keep those higher abundance targets in the 488 and 520 channels. So in terms of 3-color Western blots, the recommended workflow is, the recommended approach essentially is to use the revert 520 total protein stain in the 520 channel for normalization. And then you use your 700 and 800 channels for your targets of interest. In this case, you have BCL2 and BCL-XL in the 700 and 800 channels respectively. And the great thing about this is that using the software, you can actually quantify the total protein and your target signals on your blot for you, pretty much automatically normalize the target data and use the total protein stain to correct for lane-to-lane variation. So you can actually see in this image below, it auto detects each lane for you and actually then spits out your graph for you. And it can go as far as actually doing statistics for you. So it's a really, really streamlined process that the software actually takes you through sort of step by step. So what about 3 actual targets with 3 actual antibodies? So this is actually possible. It is a little bit more challenging because you need to find the right antibodies, but it can be done as long as the 3 targets again are within the same linear range. So here we have beta-catenin in the 800 channel, AKT in the 520 channel, and ERK1 in the 700 channel. And you can see on the right here this combined linear range. And in this case, in order to use these 3 antibodies to detect these 3 targets, you would need to load at least 5 micrograms of protein in order to effectively quantify data using these antibodies in a Western blot. So moving on to chemiluminescence. Of course, you can do chemiluminescent Western blots with the system. Here is just an example showing that we're detecting your target using the chemi channel and detecting your protein ladder using the RGB epi channel. So in this case, it's a bit of a mix between a chemiluminescent and a colorimetric Western. So for colorimetric Westerns, you can use the 630, 525, and RGB epi fluorescent channels, and you can label for anything from Ponceau, NBT, TMB, and so forth. So moving on to gel applications, you can do fluorescent protein gels. So we've got, again, the 800, 700, 520, and 488 channel in this case. So you can label anything from Kumasi to Pro to Diamond, Emerald, and Cipro, Ruby, and Orange. For colorimetric protein gels, you can use the 630, 470, and RGB trans channels. Again, Kumasi on the left-hand side here and silver stain on the right-hand side, just showing the images. And finally, the nucleic acid gels, you can use the 700, 520, and 488 channels. You can look at ethidium bromide, gel red, cybersafe, and so forth. So moving on to multi-well plate assays. You can do in-cell Westerns, of course. Now that we have these 4 channels, you've got the 800, 700, 520, and 488 again, you can do your typical 2-channel in-cell Western blot, but now you can also do this 3-color in-cell Western as well. Again, preferably keeping cell tag, in this case, for normalization in the 520 channel. And one of the things that it allows, one of the important things with the software is actually allows you to do, again, cell, look at a combined linearity again. And you want to do this again for cell tag, actually looking at cell stain linearity, and that's an option in the workflow. And part of this is related to assay development options the software provides you. So you can do anything from antibody titrations, blocker evaluations, and so forth. So it's really, really useful to sort of take you through the entire process from, developing your assay all the way through to the statistics at the end. You can do ELISAs using the 630 and 470 trans. Here you've got an ELISA at varied concentrations of programmed death ligand 1. and of course, any sort of fluorescent cell-based assay, essentially using any of these channels. Here we've just got an example of a viral titration assay, knockdown assay, a reporter gene assay. And finally, going through to the microscope slide applications. Again, we can do a 3-color image of the, in this case, pig intestine. And what I really like about this is you do get this high detail at this 5-micron resolution. So here we've got nuke spot in blue, beta-catenin in red, and nuke2 in green. And this is really useful for bulk tissue imaging and screening. Can really help you find specifically where your protein is expressed without having to do high-level microscopy 1 slide at a time. So it's a really big time saver. And then once you find where your protein is expressed, you can go ahead and do that higher-level microscopy. Personally, this is one of my favorite things because it just speeds everything up. Kind of wish I had this when I was in the lab. And finally, you can look at histology section, again, at that 5-micron resolution using the RGB trans channel. So you can look at things such as H&E stains, Elysian blue, etc., and so forth. So you can see there's a whole range of applications that you can actually do with the Odyssey M. From quantitative Western blots, where of course that validation is important. You've got the 3-color Western blot, which is possible. You do qualitative Western blots, where you're just doing sort of a quick and dirty, is my protein there, is it not there? You can do protein nucleic acid gels, in-cell Westerns, ELISA, GFP assays, cell viability, proliferation, tissue imaging. And so I'm just going to quickly take you through now how the system actually, sort of the system imaging modalities, which I briefly talked about. So I mentioned the line scanner before, allowing you to do faster scanning compared to its predecessors. So this essentially is just a line that scans, one part at a time. And this is a lot faster than the point-to-point based system because it is a line, obviously, so it can do that faster rather than doing one single point at a time. For the epi-fluorescent channel, so this would be for membrane applications. So you'd have the scan in this fashion with the detector on a moving plate. For transfluorescence, so this is for slides and plates. You have the detector on the top, again, moving with the plate. And finally, for chemiluminescence, we have a stationary plate in the top left-hand side, allowing you to do chemiluminescence in that top left-hand corner. So it's a 13 by 15 centimeter imaging area, which you can actually do your chemiluminescence on. You can also do bioluminescence on this area. And of course, throughout the entire region, you can actually do everything else. So with the Odyssey M, you can do up to 6 membranes at a time, 4 plates at a time, or 12 slides at a time. So you've got that nice throughput with the system. And so the system comes with these accessories, comes with the plate holder for the 4 plates and the slide holder for the 12 slides. And there's sort of a hidden compartment on the front of the system where these are sort of kept and slide in, along with the silicon mat for Western blots. So probably one of the most important things that actually comes with the system is the image acquisition and data analysis software package. Now, the Imperial Studio is the analysis software that comes with the system. It's a comprehensive package. And this software has actually been designed in collaboration with scientific journals and funding agencies to really get you the best quality data. As I mentioned before, you can do assay validation all the way through to statistical analysis, and it really takes you through step by step through the workflow. And then you also have the LAS software, the Lycor acquisition software, which is what essentially controls the system. So as I mentioned, Curious Studio really guides you from that validation all the way through to the statistical analysis and really gives you the most accurate and reproducible results to get that publication done. It really takes you through step by step. And that's really the power of the software, the Odyssey M. So just to quickly summarize, the primary applications for the Odyssey M imager, quantitative and qualitative Western blots, in-cell and on-cell Western assays, and tissue section imaging. So it's a leading system in fluorescent Western blots. And the combination of the M plus the Imperial Studio software really provides an unprecedented workflow analysis tool, and the higher resolution channel flexibility really enable a wide variety of macro tissue section imaging capabilities. So feel free to ask any questions if you have them. Otherwise, feel free to contact me whenever. Thank you. Back to you, Fraser.
Thanks, Anthony. Yeah, awesome presentation. Lots of information there. So we won't take questions on Anthony's presentation just because we're running a little bit behind on time, but I've just launched a poll. So if you guys could just fill that out really quick, that'd be awesome. And if you have any questions for Anthony, just pop it in the chat so he can answer them during the presentations. Our next speaker and the final speaker for this session will be Associate Professor Matloob Hussain. This is kind of like another little Millennium Roots session for me personally, because Matloob actually taught a few modules back when I was in the University of Otago doing my master's courses. So it's pretty cool to have Associate Professor Matloob speak with for us today. So yeah, I will pass on to you.
Thank you, Fraser. And I'm sorry, I keep actually replying to you with your name like Frazier. Sorry for the now. I realize actually much later on, but
thank you so much, Matloob. I've never ever received that apology before, but I really appreciate it.
Thank you for the invitation, and I'm really enjoying the session and the overall webinar. And also very interesting talks previously, and I will be really interested in this new Odyssey, because so far we have been using Odyssey FC, and I guess that's why you invited us or invited me to talk about it. And so let me share. How do I share? It says only one participant can share at a time.
How about that? Is that better now?
Yeah, how do I? Okay, now I can. I hope I don't share the wrong screen. Okay. Okay. So yeah. So this is the screen. Can everyone see it?
It's just blank at the moment. Oh no. Yep. Got it now.
Okay. Yeah. So. Yeah. So it's doing things. Yeah. So so the title of my talk is Antagonism of Antiviral Host Factor HTEC4 by Influenza virus. So basically, I'm going to emphasize on the technique and the machine we use to actually detect that or analyze the antagonism, and we use Odyssey FC Major throughout this study. And this project was done by my PhD students who actually finished a few years ago.
Sorry, Matloob, we can see the notes on screen as well. So I think if you go up to display settings, that's the way to get the full screen up.
Display settings?
Up on the top next to...
Which one? The sharing set options?
Yeah, so if you just share screen, it should pop up.
I'm sharing it right now. Is it?
No, so it's just got the PowerPoint. So I think it's in the, if you go to your upper left-hand side, where the orange like.
Yeah, no, you need to put it into presentation mode first, Fraser. So you had it right, you were in presentation mode, and now, do you have two screens?
Yep.
Yeah, so look on your notes screen, and across the top you'll see this, show taskbar, display settings, and end slideshow. Yep, keep coming up.
Yep, here.
No, for up to the top of the screen. I can see your cursor. Move up.
Oh yeah.
Yep. So now the one that says display settings.
Oh,
in the middle, click that, and then it should allow you to swap displays. Yep, just hit that one, swap presenter, and then you should be good to go. Perfect.
Okay now?
Yeah,
perfect.
Thanks,
Thank you for help. Yeah,
I do it all the time. So
I'm using my laptop and a big Samsung monitor.
I hear you.
Thank you for that. Yeah. So where was I? So yeah. Yeah, so I was saying that this project was done by my PhD student, Henry Gelvin. So before I go on to talk about the actual data, and obviously you are familiar with influenza virus, maybe forgotten about it because of the events are going on because of the SARS coronavirus 2. It's kind of like there's a competition going on between influenza virus and SARS, but remember that the phenomenon of pandemics and everything actually started by this particular culprit here. So the reason we don't see much news or cases about of influenza, one is that potentially it's not out there because of the lockdowns and restrictions and everything. And then second is actually we are actually not looking for it. You know, we are not really testing because our focus is testing on the SARS 2.0. But when you go into detail, when you look at the data which keeps coming, that there's another phenomenon is occurring during this pandemic, that there are actually co-infections of influenza virus being discovered, both in actually clinical settings as well as in the experimental settings in the lab. And the people are talking about that there might be a phenomenon kind of like twindemic, you know, might happen, epidemics of SARS and influenza together. And it could be very complex for the clinical outcomes because primarily influenza is an upper respiratory tract infection, and SARS 2.0 is a lower respiratory tract infection. So theoretically, basically both viruses can actually exist in the same individual. So that is theoretically frightening, but we'll wait and see how it goes. So when we look at the virus, so this is the virus, this virus is actually a little bit different than other viruses. It has these phenomenon, antigenic drifts and shifts, and continue to change. And one of the reasons for that is the RNA genome, which is segmented. So basically, it can actually, if you have infection from two different influenza A viruses, they can mix and match and give rise to a new influenza variant, a variant which can actually cause a pandemic. And another issue is that we continue to, because it's an RNA virus, and they use frame shifting in alternative splicing and other mechanisms to maximize the gene expression. So we continue to actually identify new genes out of that phenomenon, and these are actually highlighted in red. So we think we know everything about influenza, but I don't think we do. And even if we keep doing research, keep generating the data and knowledge, influenza continue to evolve and always stay one step ahead. And just like SARS 2.0, it continues to evolve. Okay, I don't want to digress too much. So my lab is interested in understanding the fundamental relationship between a virus and the host, because viruses are obligate intercellular pathogens, so they need a host to multiply and to cause the disease, and for that they need to get inside the cell and multiply. Okay, and when we always look from our own perspective. Viruses, dangerous and this and that. But when you look at from the perspective of a virus, when virus goes inside a host cell is like a mall. When we go to a mall and just one person go to the mall and then you enter the door and you see, wow, there's so many stores, so many people and out there. So it is that kind of environment for the virus, the host cell. And host cell itself, it's very complex. It's a microscopic structure, but inside it is so many things are going on, and some of those things are actually highlighted in this slide, particularly. So you have a genome, you have proteome, you have transcriptome, you have so many things are going on. So imagine from the perspective of the virus that as soon as the virus, enters the cell through a gate, and then it has to encounter everything what is in front of it. So the host proteome particularly is, again, it's in itself is very complex. And one of the phenomenon that actually contributes to the complexity of the proteome is the protein modifications. Means like one protein, one polypeptide can actually exist in different species, modified by different modifications. And what I mean by that is this. So the modifications are known: phosphorylation, glycosylation. We know that And then there are very common and popular modifications as we know of. But there are, in addition to that, there are so many other modifications exist, and some of them are listed here, and it's actually not an exhaustive list. So one of the modifications we are interested in is the acetylation, so that when the proteins, when that proteomic environment, which is hostile to the virus, when it is modified by the acetylation, then what kind of implications occur for the virus itself, and on the other hand, so that's the implication for the virus. But then how virus actually exploit the situation in the cell using these acetylation environment and work towards its own advantage. So when you look at the acetylation itself, the modification is actually regulated by two category of enzymes, histone acetyl transferases. So histone is just a misnomer because it was first discovered in histones. So you have acetyl transferases and you have deacetylases, adding the acetyl group and then removing the acetyl group from a particular polypeptide and creating a dynamic environment. But then when you have this imbalance between acetylation and deacetylation, and then you have then this imbalance contributes to multiple diseases and deformities, and it is known in cancer, diabetes, and diseases like Alzheimer's. So same could be true for viruses as well, because viruses rely on the host cell environment to multiply and to antagonize things which they don't like. So years ago, when I was in US, I did an experiment, and I just used a broad inhibitor, which could actually inhibit multiple deacetylases. So basically increasing the acetylation environment in the cell. And that resulted in the increased influenza A virus replication, indicating that influenza virus actually likes the acetylation environment in the cell to proliferate. So to dig deeper a little bit into it, so we are working on individual aspect of this. So from the look of it, if you think that the acetylation is preferable or promoting the influenza virus infection, then the acetyl transferases would be pro-influenza, and the deacetylases would be anti-influenza. Okay, and then influenza will exploit the acetyl transferases to its advantage, so that actually it can acetylate the host proteins as well as the viral proteins, and we have the evidence of that many viral proteins are actually acetylated in infected cells, so that influenza can actually exploit acetyl transferases to acetylate the protein and multiply efficiently. But on the other hand, deacetylases are a kind of enemy. They are going to interfere in that process, because they will take away the acetyl group from the viral proteins as well as host proteins. So and then obviously they are antiviral. So first we looked at by using a technology which is kind of running in parallel, RNA interference, and now people are using CRISPR-Cas to knock out the particular gene. We don't prefer the knockout because knockouts are the lethal. Because we have to do a selective modification of the host environment, because to infect the cells, we actually need a viable, or slightly or somewhat viable cells, so that we can infect the cells with the virus after the manipulation, and then look at the phenotype, what phenotype is there. So by using that technology, RNA interference, we identified multiple histone deacetylases. They actually have antiviral function. So the question arises that if all these deacetylases have antiviral function, then how influenza virus goes, antagonizing them and bypassing them so that they don't interfere its replication. So that's what the focus of my talk today is, that how influenza virus antagonizes the histone deacetylases, or in short, HDEX. So in general, the antagonism should occur and does occur at two levels, at mRNA level as well as the polypeptide level, because when any gene expresses, the kinetics of the mRNA expression are different than the polypeptide expression. So you have the increased expression of the mRNA, and then mRNA expression will go down, and that the polypeptide will express, and that depending on the half-life of the polypeptide will get degraded. So that's a normal process. So viruses don't do anything new. They don't bring anything new to the party unless they have to. So what they do, they basically, to antagonize any protein, in this case HTEX, they basically exploit the machinery which already exists in the host cell for housekeeping. So our interest in this case was HTEC4. So we already identified that HTEC4 has an antiviral function during influenza virus infection. So we also know that in HTEC4 mRNA is also being antagonized. But then we wanted to look at the polypeptide level, that what is actually happening at the polypeptide level. And so this slide is a bit busy, but what it tells that doesn't matter what combinations or what strain or what subtype of influenza virus you use or how many infectious units you use, there is a downregulation of HTEC4 polypeptide in infected cells, indicating that influenza is trying to downregulate the expression to minimize the antiviral function of HTEC4. And this red, oops. So this red box highlights that there is a downregulation, and so we use the Odyssey FC imager, and we also use chemi labeling and what Anthony was describing earlier, and also fluorescent staining and combinations and everything to see to measure the downregulation, and we use different loading controls. So you have a loading control here, actin, and then you have a marker of the infection, and P, and you can see that there is a definitely downregulation of HTEC4, a particular condition, and then we use an inactivated virus here to see that this is actually because of the infection, not because of the other effects. And then we can actually quantify what Anthony was describing. So in this case, we use Imager Studio software, and we can quantify that how much reduction in the polypeptide level is there because of the different conditions of the infection we are using. So here, by using these different conditions, we can actually say that HTEC4 polypeptide is actually being downregulated in infected cells. So what is the mechanism? So then we did the time course that what time of the infection the polypeptide is actually downregulated. So it's occurring sometime between 12 and 24-hour infection. But as you see that the full length is decreasing, at the same time that few other bands are popping up at the bottom. One is here and one is here. So this indicates that this is occurring through the cleavage. So basically the protein is being chopped up into smaller fragments during the course of the infection. So we had some idea why this is the case because of this. So this is a viral protein. This is also get cleaved into two fragments by caspase. Caspase, which is a series of group of enzymes which take part in the programmed cell death called apoptosis, and virus infection is known to induce the apoptosis. And so at the same time with this labeling, we also use caspase 3 antibody to stain for caspase. What is happening with the caspase? Caspase is actually expressed as procaspases. And during the process of the apoptosis, they have to be activated by either self-cleavage or cleavage by another caspase. So here you have a procaspase during this the course of the infection. So this is a full length, and then around this time, this caspase 3 is also getting activated with this cleavage. So we got an idea from this data that it is actually caspase 3, which is actually inducing the downregulation of HTEC4 polypeptide during the infection. So next we want to solidify that and further confirm that. So we use different inhibitors. So basically there are three pathways in the host cell which contribute to the housekeeping vis-a-vis polypeptide level. So there is one lysosomal degradation pathway by cathepsins, and then you have a proteasomal degradation pathway by proteasome, and then you have apoptotic degradation pathway mediated by caspase-3. So we use inhibitors of all these three pathways to confirm that if this is actually indeed an apoptotic pathway, not other two pathways, which are actually contributing to the downregulation of HTEC4 polypeptide. And you can see here that when we use the caspase-3 inhibitor, there is a significant rescue, which is from the quantification of the HTEC4 polypeptide after the inhibition of caspase-3. And then, so this is obviously chemical inhibition. We wanted to use the genetic tool and see whether the caspase-3 is actually indeed involved. So then we knock down the expression of caspase-3 gene itself, and this knockdown, and you can see there's rescue of HTEC4 polypeptide when the caspase-3 gene is not expressing, and there is a shift in the cleavage pattern. So this also indicate that caspase-3 alone is not doing this. There are other proteases also involved in this cascade. So with the help of Odyssey, we could actually, because of the dynamic range, so you could actually detect the full length very easily, but it's very difficult to detect these smaller fragments in a normal equipment. So because of the dynamic range, we could actually detect these smaller fragments, and that's why it's a bit dirty, because it's overexposed. When you can look at it here, it's very clean. So yeah, so these slides conclude that caspase-3 indeed is involved in the HTEC4 downregulation. Then, so that's the host side. So influenza is exploiting the host machinery to knock off a factor which is antiviral, is against it. So then, which gene influenza using it to do it? So influenza basically has 8 genes, which I showed you in the second slide. These are main gene segments, but they can encode more than one protein. So, so 2 of those genes express the surface protein HA and NA, which are the main antigen. But the 6 of those genes are internal. They express polymerase subunits, and then other component like viral core protein, which helps in the replication and all that. So we were not interested in the 2 genes, HA and NA, because we were not suspecting that they would actually induce the downregulation of HTEC4 polypeptide. So again, we utilize the RNA interference and individually knock down the expression of these 6 genes, and 4 are shown here: PB1, PA, NS, and NP. And so when we knock down all these 4 genes, we don't see the rescue. But when we knock down we knock down 3, we don't see the rescue. But when we knock down PA, we see a significant rescue of HTEC4 polypeptide in the infected cells, and slightly in PB1, but it's not as significant. So this slide, again, with the help of the Western blotting images visualized by Odyssey FC and quantified, that indicates that PA gene of influenza virus is actually involved in downregulating HTEC4 polypeptide in infected cells, and when we knock it down, there is a rescue of HTEC4 polypeptide. Okay. So then next, as I said, that the viral many influenza genes, they express more than one polypeptide. So PA gene itself is this here. It can actually encode multiple other variants, and one of them is called Pax. So it is generated by a ribosomal frame shifting. So Pax has an endonuclease domain at the N terminus here. This one. So this is the full length PA, and then this is Pax, which is actually encoded by PA gene by ribosomal frame shifting. And this protein has the whole endonuclease domain, and then this actually chops up the RNA, host RNA during the infection to antagonize multiple proteins. So what we did here, we got a plasmid which is encoding just the Pax, and then we express that in the cell without infection and see whether Pax itself can actually induce the downregulation of HTEC4 polypeptide. And you can see actually it does. It can actually induce that downregulation of the HTEC4 polypeptide. So basically, what I showed you here, the data of a project which we wanted to know whether HTEC4 is an antiviral factor, and it is, and how influenza virus is antagonizing the HTEC4, and what equipment and what methodology we use to do that. And in the broader term, it's actually very important to know the nuts and bolts of the machinery which is involved in influenza virus infection, and how influenza virus is actually, unscrewing or screwing those nuts and bolts. So that is, in a bigger term or bigger picture, when you look at the host, acetylation machinery plays actually an important role during influenza virus infection, and could be actually a novel antiviral targets. And acetylation is being targeted in other diseases like cancer and Alzheimer. So some of those drugs actually could be repurposed for this purpose as well. But again, including us, there are only few groups working on this side of influenza virus research, and so it is also still in a very very much infancy, and we are trying to understand the role of individual HDAC or HATs, that is acetyltransferase, in influenza virus infection, to get the bigger picture, the whole picture, that how this interplay is occurring between virus and the host, so that we can move on and try to develop some antiviral drugs. Thanks. Sorry, the acknowledgements.
Awesome. Thank you very much, Matlub. That was a fantastic presentation. And yeah, it's good to kind of hear about something that's not Corona for once.
Yeah.
Music to my ears. We will have some time for some questions, maybe just one or two. So if you guys could, yeah, raise your hands or